Okay, so I got cut off in the last video. Welcome to part 3. Other biochemical tests can be done to detect other genetic disorders. So karyotyping is just not the only option that's available. Other tests can also be done of the chemicals in the amniotic fluid and from the cells. Okay, so let's see, chorionic villus sampling. This one is a less invasive and a sample that can be done earlier during fetal development. So what they do is they insert a suction tube through the cervix and they take the cells that are part of the chorionic villi, which would include some of the same fetal cells. So they can do the same thing. They can take those fetal cells and they can karyotype them, or they can do biochemical tests to determine whether they have certain genetic disorders. So this one is less invasive. It's a little bit safer than the amniocentesis, and it can be done earlier. Okay, and we talked about this already, that some genetic disorders can be detected at birth, and we routinely do these tests in the United States. They will poke an infant's foot and get a blood sample on a card that's sent to a lab, and the lab in the hospital will test infants for certain diseases, genetic diseases. Okay, so I wanted to review some pedigree charts. I said we would be doing some pedigree chart problems. So I wanted to look at this one. Does this one look like it's a autosomal or sex-linked? And we haven't talked about sex linkage yet, but we will. Does this look like it's autosomal? Does it look like it's sex-linked? Is it dominant or recessive? Okay, so right away we know that these people have the trait. So Arlene in the first generation, she has the trait. So does Tom. Wilma, Tom's sister, has that, and so does Carla down here, Tom's niece, and Arlene's granddaughter, Carla, has that trait. None of these other people have that trait. So right away, the kicker here that we know that this is a recessive, autosomal recessive trait is Anne and Michael. Anne and Michael do not have that trait. So therefore, and before we say therefore, Carla, their child, does have that trait. Therefore, Anne and Michael are considered carriers for that trait. So they are heterozygous. They would have one big letter and one little letter for their genotype. And the only way that Carla can have that is if they both are heterozygous. So Carla has two little letters for her genotype. Now, we can also determine that everybody who has this has two little letters for their genotype. They are homozygous recessive. Okay, what about George? Well, we know for sure that George is heterozygous for this trait because George has children that have this disorder, Tom and Wilma. It might not be a disorder, but we know that Tom and Wilma have this recessive trait. Okay, the only way they could receive that is one allele through Arlene and one through George. And since they have those traits, George has to have a little letter in his genotype. He's got to be heterozygous. Okay, Sam is also heterozygous. We know that for sure because he does not have the disorder, so he has to have at least one dominant gene. And Arlene, the only thing she can give him is the recessive gene. So we know that Sam and Anne are both heterozygous. Okay, Sandra and Tom have Daniel and Alan. Alan and Tina have Christopher. So we may be tempted to say, all right, Daniel and Alan are homozygous dominant, and all the rest of these people are homozygous dominant. We don't know that for sure, but it's probably pretty likely. It's more likely that they are homozygous dominant than not. But further generations would have to be gone through to determine for sure whether Daniel, Alan, Tina, and Christopher are homozygous dominant, or if Sandra was homozygous dominant as well. So that's an example of a pedigree chart, and I could ask several questions about that. I could ask you whether it's a autosomal dominant or autosomal recessive disorder. I could ask you if it's sex-linked. So let's take a look at our final topic here today, sex-linked inheritance. And before we do that, we're going to talk a little bit about Thomas Morgan. Thomas Morgan was an embryologist, so he studied embryological development, and he experimented with fruit flies. He used fruit flies. They were good test subjects because you could produce a lot of fruit flies in a short period of time. They are easy. They have lots of different traits, and they're easy to take care of. So they're not as tricky as humans or larger mammals. And so test animals are usually ones that produce a lot of offspring and that are easy to take care of and small. So things like lab rats and worms and fruit flies make good models. And since we share a lot of genes with fruit flies, they make good models for studying human genetics as well, because we do share a lot of the same genes as fruit flies do. Okay, so I talked about those. The other reason is that fruit flies only have 4 pairs of chromosomes, so that makes it really easy to study inheritance if they only have 4 pairs of chromosomes. So their 2n is equal to 8. Diploid number equal to 8. Okay, so here we get into some complicated terms for genetics. Morgan noted that wild type or normal phenotypes were common in the fly populations. So the more common ones were called the wild type. Alternate traits are called mutant phenotypes. Okay, so things that are different from the wild type are called mutant phenotypes. So let's take a look at an example. This is the more common trait in fruit fly eye color. So this red eye color, and the white eye color is less common. So this eye color is called the wild type, and this eye color over here is called the mutant type, since it's less common. So what Morgan did is, let's look at his experiments. In one experiment, he mated male flies with white eyes, so the mutant type, with female flies with red eyes, the wild type. So just like you would probably predict, if this is a simple dominant and recessive behavior, then you would predict a crossing of true breeders to produce all red eye offspring. And indeed, that's what did happen. So far, it follows Mendel's rules. The surprising thing happened with the F2 generation. So when you take the hybrids, and when he bred the hybrids, the F2 generation showed that typical 3-to-1 ratio that Mendel got. So for every 3 red-eyed individuals, there was 1 white-eyed individual. However, only males had white eyes in this cross. Now, Mendelian genetics would predict that females and males would show the same ratios of 3-to-1. So you would expect some females to be wide-eyed, but in this case, none of the females were wide-eyed. So what does that tell us? Well, that tells us that the wide-eyed mutant allele must be located on the X chromosome, since only females can have X chromosomes. So right now is probably a pretty good time to talk about the chromosomal basis of sex or gender. In humans and other mammals, there are 2 varieties of sex chromosomes, a larger X chromosome and a smaller Y chromosome. So only the ends of the Y chromosome have regions that are homologous with the X chromosomes. SRY genes on the Y chromosome codes for the development of testes. So only males have the Y chromosome. So this is a good photograph of the X and Y chromosome. You can see the X chromosome is much larger than the Y chromosome. Okay, so this is what happens in humans and mammals. It's the same thing that happens in fruit flies. Fruit flies have this same gender inheritance pattern. Females are XX and males are XY. Each egg contains an X chromosome, while sperm cells may contain either an X or a Y. So if you do the Punnett square out, you can do that in your notes if you want to. But if you do that, you would put XY on the top for the possible sperm cells and XX on the side for the possible egg cells. And when you cross them in your Punnett square, you'll find that each offspring has a 50-50 chance of being either male or female. Now, other animals have different methods of sex determination. We're not going to get into those. Right now, we're just going to worry about how it works for fruit flies and humans. And let's go back to Thomas Morgan's experiment. So here, Morgan determined by his results that the wide-eyed mutant allele must be located on the X chromosome. And we're going to look at how that works. What he found is going to support the chromosome theory of inheritance. So even though Mendel didn't know about chromosomes, we now know that chromosomes are what carry genes and pass on traits. And Morgan's experiments supported this idea. Okay, so here we have the experiment. Here are the parental generation. So we have red-eyed females, the wild type, and wide-eyed males, the mutant type. All offspring, regardless of whether they are male or female, had red eyes. No surprise there. That was the same as in Mendel's pea plant experiments. But the surprise comes in the F2 generation. The ratio isn't any surprise, 3-to-1 ratio, which shows simple dominant and recessive behaviors. But the surprise is that only males are wide-eyed again. Okay, we saw this earlier, or we read this earlier, but here it is. The males, only the males are wide-eyed. So if it was following normal Mendelian genetics, then the 3-to-1 ratio would also apply to the females, and it would not be only males that were wide-eyed. This is Morgan's conclusion. Okay, so here we have the chromosomes, and the X chromosomes in red here denote the chromosomes that the females gave. So this is a double wild type female, and wild type is dominant in this case. And you can see they kind of write it a little bit differently than we're used to. So W is for wild type plus, means that that's dominant. So W plus denotes wild type dominant. Over here, W without the plus denotes the mutant type, or the recessive type. It's a little bit odd, but that would be the same thing as a capital R for red eye, and this would be the same thing as a lowercase r for wide eyes. So these are the only possible egg that can be produced is a wild type egg, an egg that has the gene for red eye. The sperm that can be produced are the mutant type sperm, and these are X chromosomes, right? And the Y chromosome. The Y chromosome does not carry the gene for eye color in fruit flies. It is only carried on the X chromosome. So the F1 generation, no surprise there. They are hybrids. The females are W plus and W, so they are heterozygous for eye color. And the males are W plus, and since the Y chromosome does not carry the gene for eye color, they are all red-eyed. Now when we cross this hybrid female with the male who has red eyes, these are the combinations that we get. So this male is able to produce X chromosome in one sperm with the gene for red eye, and a Y chromosome that lacks the gene in the other eye because the Y chromosome does not